Okay, good morning, everyone. My name is Jess, and it is my pleasure to be the moderator for this session titled Social Neuroscience, Emotion, and Motivation, All the Things. Okay, we have 6 speakers this morning, so we're going to run a very tight schedule. Each speaker has 12 minutes. If there is time for a quick question in between each speaker, either on the app or at the microphone, we'll see how we go, but very likely it'll be necessary to hold your questions until the very end for the panel. Okay, and on that note, I want to introduce our first speaker, Veronika Devika, the Organization of Shared Neural Substrate of Autobiographical, Semantic, and Social Cognition.
Thank you. Hello, everyone. Today I will be talking about the neural correlates that are shared between autobiographical, semantic, and social cognition. So by social cognition, I'm referring to the set of neurocognitive mechanisms that allow us to understand others and interact with them, including inferring their unobservable mental states, which is sometimes called theory of mind. The reason why we want to look at how the autobiographical and semantic memory systems might contribute is because both our memories of past experiences as well as our generalizable knowledge about the world can provide us with useful information that we can draw upon to make inferences about those around us and navigate the social world. And this is not a unilateral or unidirectional relationship because our social experiences are embedded within our past memories of past experiences, and they also inform our understanding of the world around us. Despite this intertwined nature of these 3 cognitive domains, we still don't have a full understanding of the substrate, the neural substrate underpinning the interaction between them. And this is exactly what we wanted to explore in this study. Specifically, we set to answer 3 related research questions. The first was whether tasks that probe autobiographical memory, semantic memory, and social cognition even recruit overlapping brain regions. And the idea here was that if these 3 cognitive domains do indeed rely on shared cognitive mechanisms, then they should also elicit some overlap in their neural activation patterns. To test this, we conducted 3 separate meta-analyses on the 3 cognitive domains of interest. This allowed us to identify the neural correlates reliably associated with tasks probing each of these across the entire literature, irrespective of the idiosyncrasies of specific fMRI studies. In the next step, we compared them. So we did a conjunction and formal contrast analysis that revealed the existence of overlap in 3 specific brain regions highlighted here by the black circles. So this finding was in line with our prediction and is indicative of reliance on shared cognitive mechanisms. But interestingly, in addition to all those regions, we found multiple regions that seemed to show more convergent activation for one of the domains versus both of the other 2. What this highlights is not only the unique demands of each cognitive domain, but also the potential of these regions to make unique contributions to our ability to navigate the social world. This has drove us to ask our second question, which was whether these brain regions that seem to prefer one domain versus the other 2 are functionally connected. We reasoned that if the integration of autobiographical, semantic, and social cognitive processes is functionally meaningful, then these regions should exhibit significant functional connectivity at rest. Resting state data provides an insight into the intrinsic organization of the brain, so therefore like a propensity of these regions to co-activate. We used task-free fMRI data from 154 healthy young adults. A key methodological detail here is the use of multi-echo data that allows us to have good signal-to-noise ratio across the entire brain, including ventral temporal regions that are typically problematic and suffer from signal dropout. Our analytic pipeline included extracting the time series of the parcels from the Schaefer parcellation. Critically here, we used an individualized parcellation approach that accounts for inter-individual differences in the functional organization of the brain. Then we identified the whole brain functional connectivity patterns associated with each of our domain-specific regions across participants. And in the last step, we compared them, and specifically, we computed a conjoint functional connectivity map in which the value of each parcel indicates the proportion of domain-specific regions with which it is functionally connected at rest. We found that all our domain-specific regions were functionally connected at rest with this left angular gyrus area, as well as medial and ventral portions of the anterior temporal lobes. So these regions are affiliated with the default and limbic networks in the EO7 network parcellation. This demonstrates that the intrinsic functional architecture of the brain includes a set of regions that can mediate the interaction between autobiographical, semantic, and social cognition. Next, we wanted to explore whether this network might include clusters of regions that seem to be more functionally connected with themselves relative to the rest of the shared functional network. We performed hierarchical clustering analysis, which revealed the potential existence of 4 separate clusters that had significantly different functional connectivity patterns not displayed here. What I'm showing here are the top 5 functional terms associated with activation within each cluster across the literature, as indicated by meta-analytic functional decoding. And this indicates some association of the blue cluster with perceptually driven processes, yellow with memory, purple with evaluative and affective processes, and red with both social and memory processes. So the shared functional network we identify seems to comprise clusters with different functional profiles, highlighting their potential to make complementary contributions to social navigation. We next wanted to examine the relative importance of regions within the shared network, and we did that by computing degree centrality, a graph theory metric that represents the sum of the strengths of a parcel's direct functional connections. This analysis showed that regions within lateral and inferior-medial aspects of the anterior temporal lobe, as well as medial prefrontal cortex, had the highest centrality values, suggesting that they might act as key hubs for integration across autobiographical, semantic, and social cognition. Okay, so having identified a set of regions whose functional connectivity profiles are well suited to support integration of our domains of interest, it's interesting, but it's also based on resting state data. So in the next step, we wanted to directly test whether the activation of the shared network is modulated by both social and memory-related cognitive demands. The prediction here is that if the shared functional network supports the integration of memory and social cognition, then it should show an activation pattern that's sensitive to both stimulus socialness and prior knowledge. We used multi-echo task fMRI data from a subset of 31 healthy young adults who in the scanner saw faces and places. Some of them were famous faces and well-known landmarks. Others were anonymous faces and unknown places. Participants simply had to categorize each stimulus as a face or a place. So this manipulation allowed us to concurrently manipulate both the socialness of the stimuli as well as their association with prior knowledge. We found that throughout the shared functional network, there were regions who increased their activation in response to faces compared to places. At the cluster level, it was the third cluster in the medial prefrontal cortex that seemed to show the most kind of widespread preference for faces. We also found regions that show increased activation for famous relative to anonymous faces, and this was found in both this medial prefrontal cluster as well as the ventral and ventrolateral aspects of the anterior temporal lobes. So altogether, the modulation of activation by both stimulus socialness and prior knowledge supports a role of this shared functional network in the integration across memory systems and social cognition. So in summary, we found that tasks probing autobiographical, semantic, and social cognition recruit overlapping brain regions, that the brain regions preferentially engaged by autobiographical, semantic, and social cognition are functionally connected, and lastly, that both social and memory cognitive demands modulate the activation of this shared functional network. So altogether, we provide converging meta-analytic heuristic state functional connectivity and task-based activation evidence supporting this shared neurobiology of autobiographical semantic and social cognition. And within this shared neuroanatomy, we find an important role for the default network in line with previous literature, as well as the limbic network, and particularly these ventral and ventral inferior anterior temporal lobe regions, having a very important key role across several of our analyses. And what this highlights is the importance of employing fMRI acquisition and preprocessing pipelines that can detect bold signal in this region in order to arrive at a comprehensive understanding of the neurocognitive architecture of social cognition. And I want to thank my supervisor, Nathan Sprang, the Laboratory of Brain and Cognition at the Montreal Neurological Institute, our collaborators and funders, and of course, you for listening.
We have some time for questions. I'll ask a quick question then. With your famous versus your anonymous faces in your task-based fMRI experiment, did you verify that those were famous faces to your subjects?
Yes, we had a post-scan manipulation check in which participants rated each stimulus on a Likert scale based on the degree of familiarity. And we only included faces that confirmed or were rated by the participants as famous in our analysis.
And same with the places?
Yes, yes.